Hey, this is Group L, and today we will be talking about Kabuki Syndrome and how it relates to the KMT2D gene. It is worth noting that there are two gene mutations known to cause Kabuki Syndrome, genes KMT2D and KDM6A, but we have opted to discuss the former because mutations in this gene are more commonly tied to Kabuki Syndrome. So, hopefully by the end of this lesson, you will be able to define what Kabuki Syndrome entails, meaning common symptoms of the disease and its cause. You should also be able to describe inheritance patterns for the KMT2D mutation, describe the structure and function of the gene, transcript, and protein, and to relate KMT2D mutation to the symptoms associated with Kabuki syndrome. We will also discuss attempts to target KMT2D therapy. One of the most readily visible symptoms of Kabuki syndrome is the way it causes improper development of the shape of the head. The disease is named Kabuki because not only was it discovered in Japan, but also the head shape is reminiscent of the masks worn by traditional Kabuki actors. Note in particular the everted lower eyelids, the broad nose with a flattened tip, prominent eyelashes, and arched eyebrows. Additional facial features include blue sclera, drooping of the upper eyelid, misaligned eyes, a highly arched roof of the mouth, depressions of the inside of the bottom lip, and an abnormally small jaw. Kabuki syndrome is a relatively rare disorder, occurring approximately once in 32 to 86,000 individuals. Aside from the head deformations, there are other common symptoms in other parts of the body, making the KMT2D gene pleiotropic. Microcephaly, seizures, and intellectual disability are tied to improper head development, but Kabuki syndrome patients are also known to have heart defects, hypotonia, and malabsorption. The KMT2D gene is located on chromosome 12 on band Q13.12. This makes it an autosomal gene, from which you can infer more about its inheritance pattern. So here is an example pedigree of how KMT2D mutations might be inherited, with red being a diseased individual and blue being a normal individual. Of course, since we clarified it is on chromosome 12, making it an autosomal gene, males and females are equally likely to inherit the condition from a diseased parent. Furthermore, although mutations in KMT2D are loss of function, the KMT2D gene is haploinsufficient in heterozygous individuals, so the inheritance pattern is actually dominant rather than recessive. The disorder does not seem to affect a patient's lifespan in most cases, although in some cases KMT2D mutations can lead to abnormal gonadal development, such as cryptorchidism. Kabuki syndrome patients are still expected to have normal fertility, making this pedigree a reasonable approximation. Here are two different views of the KMT2D gene transcript, which has a total length of 54 exons and 19,419 base pairs. Jagged lines represent introns, boxes in the top view and vertical pink lines and boxes in the bottom view represent exons. Alternative splice variants are shown under the top strand in pink. There are so many because it is such a large gene. So, after the transcript is translated into a protein, here's what you get. Again, since the gene is so large, there are quite a few functional domains. Most noteworthy are the ones marked here. The 7-plant homeodomain, or PhD zinc fingers, are required for recognition of lysine methylated histone H3. The high mobility group HMG box functions in DNA binding. The FY-rich domain, N-terminal and C-terminal FYRN and FYRC are sequences common in histone H3 lysine group 4, aka H3K4 methyltransferases, but are poorly understood. Finally, the SET domain is the most important functional domain on this protein, which methylates H3 on lysine 4, so it can be considered the primary active site. All of these protein domains are highly conserved between many species, signifying their importance in gene regulation. So essentially, what all of this means is KMT2D functions as an H3K4 methyltransferase, methylating the lysine 4 position of histone H3. These H3K4s are important in marking promoters and enhancers for many genes, and KMT2D is particularly important for binding enhancers. KMT2D, along with a variety of other regulatory proteins, are needed for transcription of specific genes and is particularly important in maintaining somatic cell identity. Mutation leads to an inability for RNA polymerase II to properly transcribe genes, sometimes causing pausing, stalling, and backtracking within a gene. And of course, because the gene is haploinsufficient, even in heterozygotes for a loss-of-function mutation, you still aren't getting enough proper transcription. So, where in the body does the KMT2D mess with the transcriptional machinery of these cells? It gets expressed mostly in the bone marrow, the cranial region, the heart, and in adipose and muscle tissue in adults. But actually, this is due to where these cells originate from in development. Early in development, transcriptional signals lead to the differentiation and migration of neural crest cells to become somatic cells. However, when the KMT2D gene is unable to properly transcribe the genes, these neural crest cells can't go where they need to go or become the types of cells they need to become. This is why all these cell types that result from neural crest cells end up getting affected. Being expressed primarily in bone marrow and the brain is consistent with the idea that Kabuki syndrome leads to improper development, particularly in a cranial region. It is also shown in adipose and muscle tissue, which would explain the hypotonia and malabsorption. Finally, improper development of the heart leads to septal defects and coarctation of the aorta. At the moment, especially because of how recent this disorder was discovered, only palliative care for managing symptoms of the disorder exists. Although, as stated before, Kabuki syndrome is not known to lead to a decrease in life expectancy. A potential treatment could be administering either the correct KMT2D protein in areas of the neural crest during the right times of development or some other similar protein capable of performing methylation. However, this would be extremely difficult as it would have to be done very precisely during the correct stages of embryonic development in the proper location. Alternatively, papers have shown that KMT2D is partially functionally redundant with KMT2C. If KMT2D is non-functional, doctors could instead signal for increased expression and activity of the 2C gene and perhaps restore proper methylation. So, overall, I hope that by the end of this lesson you have learned what Kabuki syndrome entails, meaning common symptoms of the disease and its causes. You should also be able to describe inheritance patterns for the KMT2D mutation, describe the structure and function of the gene, transcript, and protein, and to relate KMT2D mutation to the symptoms associated with Kabuki syndrome. You should also have at least some idea of how KMT2D mutations may be targeted for therapy. Thank you for listening.